The goal of this video is to illustrate how hippocampal slice cultures can be used to decipher the neural immune signaling that modulates susceptibility to spreading depression, the most likely underlying cause of migraine aura and perhaps migraine headache. Spreading depression is easily evoked in hippocampal slice cultures. Since the cultures are maintained in vitro for weeks, the behavior of individual cell types can be measured, mimicked, and modulated over extended periods. This is especially important for the study of neural immune signaling. Since mature hippocampal brain slices attain a quiescent immune state, immune responses of spreading depression can be assessed without the potentially confounding effects of anesthesia. Using modern molecular biological techniques, sensitive detection of cytokine signaling from as few as 50 cells to approximately 90,000 cells in a slice can be determined. Accordingly, hippocampal brain slice cultures are ideally suited for use in the development of novel neural immune therapeutics to alleviate migraine.
My name is Yelena Grinberg, and this is Pusic and Heidi Mitchell from the laboratory of Richard Kraig and the Department of Neurology at the University of Chicago Medical Center. Today we will demonstrate the use of rat hippocampal slice cultures in the study of low-level immune signaling of spreading depression. Spreading depression is a paroxysmal neural event that's triggered by a burst of increased synaptic activity that's then quickly followed by a transient loss of all activity. Increasing evidence shows that low-level immune signaling can influence and in return be influenced by synaptic activity within the brain. We hypothesize that cytokines modulate susceptibility to spreading depression, and as a result, migraine aura and perhaps migraine headache as well. The procedures we describe here are designed to minimize potentially